In today's video, we're going to explore the groundbreaking work of Gregor Mendel, who is widely regarded as the founding father of the field of genetics. Mendel was an Austrian scientist and monk who lived during the 19th century in the 1800s. Although farmers had already known for thousands of years that crossbreeding the strongest or healthiest plants could lead to better and more productive offspring, nobody at the time understood why or how it actually worked. While living at his monastery, Mendel began conducting detailed experiments on pea plants in the garden, where he specifically observed how certain characteristics, such as plant height, flower color, and pod color, were passed on from one generation of pea plants to the next. To better understand Mendel's findings, let us look at one of his key experiments. In this experiment, Mendel selected one pea plant that had green pods and another pea plant that had yellow pods, and then he crossbred these two plants. Interestingly, all of the offspring resulting from this first cross had yellow pods. None of them were green. But the story does not end there. Mendel then took two of these yellow pod offspring and crossbred them with each other. This time the results were different. 3 out of every 4 plants had yellow pods, but 1 out of every 4 had green pods. At first, this result might seem puzzling. Why would crossing a green plant and a yellow plant produce only yellow offspring, while crossing 2 yellow plants leads to the reappearance of green pods in some of the next generation? Mendel's conclusion was that some factor must be passed from parents to offspring, and he referred to these as hereditary units. Today we call them genes. More importantly, Mendel observed that some of these hereditary units could be dominant while others were recessive. A dominant hereditary unit would always be expressed if present, while a recessive one would only be expressed if it was inherited from both parents. In the case of his experiment, the yellow pod trait was dominant, and the green pod trait was recessive. That meant that in the first generation, the green pod trait was still present in the genetic makeup of the yellow offspring, but was not visible because the dominant yellow unit overpowered it. However, when 2 of these yellow plants were crossed, some of the offspring inherited 2 copies of the green unit, and so the recessive trait was expressed again. Mendel repeated similar experiments, focusing on other traits such as flower color and plant height, and again found consistent results. Each trait seemed to be inherited independently in a pattern involving dominant and recessive factors. This led Mendel to believe that many different characteristics were inherited in a similar way. It is important to take a moment to understand the historical context. All of Mendel's experiments and observations were made in the 1800s, which was nearly 2 centuries ago. During this time, scientists had no understanding of DNA or the existence of genes as we define them today. Because of this, Mendel's work went largely unnoticed for many years, and it was not until decades after his death that the scientific community began to appreciate the significance of his discoveries. Toward the end of the 19th century, researchers discovered chromosomes and started observing how they behaved during the process of cell division. In the early 20th century, scientists began noticing parallels between the way chromosomes were inherited and the hereditary units that Mendel had proposed. This led to the realization that these hereditary units, now called genes, were located on chromosomes. The field of genetics saw further major advancements in the mid-20th century. In the 1950s, the structure of DNA was discovered. Scientists identified that DNA has a double helix structure, which provided even deeper insight into how genetic information is stored and transmitted. The most recent major milestone occurred in the year 2003, when the Human Genome Project was completed. This achievement meant that scientists had successfully mapped the entire sequence of the genetic bases in human DNA, allowing us to understand the complete code that makes up human life. And that brings us to the end of our look at the origins and history of genetics, starting from Mendel's humble pea plant experiments all the way to the modern-day sequencing of the human genome. It is truly amazing how one monk's work in a garden helped lay the foundation for an entire scientific discipline that continues to evolve and shape our understanding of life today.